Okay. Thank you, everybody, for bearing with those technical difficulties. So I'm gonna share with you guys a few highlights of Emilie's background. We have a really qualified person coming to speak with us tonight about our subject. So Emilie is a senior endangered species conservation biologist, freshwater mussel lead for the Xerces Society based in Portland. She holds a master's of science in biology from Kansas State University and a bachelor's in biology from Berea College. Her professional experience includes more than 10 years working in biological research, conservation, and restoration. And her recent work ranges widely from surveying for high alpine stoneflies, fantastic, to snorkeling rivers to observe freshwater mussel populations. And in her current role, she also serves as the lead on freshwater mussel conservation work at Xerces. So thank you very much for making the trip.
Thank you all for having me. It's always a pleasure to talk about mussels, and I'll just point out in this picture, everybody sees the fish first, but it is in fact hanging out next to a western pearl shell. And this is just a nice example I like to start out with. You see really nice riparian roots behind and good burrowing gravel. And, just a nice example of freshwater mussels thrive in really healthy stream habitat. All right. So just briefly, most people who know about Xerces are familiar with our work on pollinators. That's what most of our staff work on, whether it's monarchs or bees or other insects as well. But we do have projects that focus on other vertebrates, including some of the rarer or less appreciated ones. So that includes high alpine stoneflies, for example, but also freshwater mussels. So our program is focused out of the Portland office, although I'm local to the gorge. And our work focuses on species of freshwater mussels that are found in the Western United States. But we are broadly focused on freshwater mollusk conservation efforts, around the country. So just as a nice way to place mussels in context, none of these are mussels, but they're all mollusks. So these are creatures that are closely related to freshwater mussels that people are often more familiar with. So the octopus, the snail, the banana slug, the giant clam, the fingernail clam, and marine mussels on the coast are all mollusks. And so they share some important characteristics that help us understand how freshwater mussels live and exist in our rivers and streams. So mollusks are incredible. They're the second most abundant species groups of invertebrates after arthropods. They can be tiny, they can be huge. They have really cool differences between their adult stages and larval stages. So a baby octopus, looks different than an adult. A baby snail may look very different than the adult. A baby marine mussel might also look quite different than the adult stage because they have these different periods where they may exist more along the bottom or the intertidal zone and other periods where they might be more pelagic or floating in the water. What binds them all together is that they have mantle tissue, which is what secretes their shells. Some mollusks have lost their shells, but that mantle tissue is essentially what makes them a mollusk. So thinking about freshwater mussels, the image on the far right is a western pearl shell again. And I think this is a nice example because what you can see here is the foot is on that left side burrowing into the bottom of the stream. This is an important feature of freshwater mussel that differs from a blue mussel because blue mussels glue themselves to rocks with byssal threads. Whereas freshwater mussels, they don't use a byssus as an adult. They actually have to wiggle themselves down into the bottom. And that plays a role in how they interact with our stream. You can also see there, this is actually the mantle tissue exposed here. So this is the part of the mussel that's secreting the shell. And then a mussel is characterized by being a bivalve, having two valves. And then you can't see it quite from this angle, but they have two openings, an in-current opening and an ex-current opening that assist them in filtering. The other species that you see here in the middle, the zebra and quagga mussel, that's what most people here or are familiar with when you say freshwater mussel. So it is interesting when I start a conversation, I have to say, but not the bad ones, because zebra mussels are highly invasive, as we know. It's a concern about their introduction into our rivers and reservoirs. But they have a very different life cycle than our native species. And they just live very differently by gluing themselves to the infrastructure. Asian clam, that's Corbicula fluvidia. This is a very common invader in our rivers and streams across the state. It's believed that it was first introduced into North America through the Columbia system. They've been here for a very long time, many decades at this point. They're widespread. They look kind of superficially like a freshwater mussel in that they have those two openings. You can see a little incurrent and a little excurrent in this picture. But they tend to be sort of epibenthic on top of the sediment, sometimes burrowed in. But they don't use a host fish, which is the key difference between them and freshwater mussels. So I'll get into that in another slide, but they are widespread. They seem not to have an effect on our native species, but it's possible where they reach really high abundance, they could be having an impact.
Will you explain what it means to be a host, have a host fish?
I
instead
will,
of-
yeah.
Okay.
Yep, I have some really fun videos. Yep.
Cool.
Yeah, so this is not the video, this is the diagram part, but this is a nice way to just kind of see it all at once. So if you're a freshwater mussel, you could be a male, you could be a female, or you could be a hermaphrodite, where you can produce both eggs and sperm. In the case of our western pearl shell, sometimes a single mussel bed will have a mix of males, females, and hermaphrodites. So 5% could be hermaphroditic, and then the rest would be one or the other. They are really long lived. So when you see them in their adult stage burrowed into the bottom of the stream, they could be 100 years old. It's common for them to reach ages of 40, 50, 60, if the habitat is good. Our other mussel species that I'll point out as well, they're long lived too, relative to a lot of the other freshwater macroinvertebrates that we know. So they can live 10 years or 50 years or 60 years as well. But this is our really long lived one. And actually closely related species in Europe can live over 200 years. So really incredible that they have these long lifespans. And we know how old that they can get by the lines on their shells. So they deposit annual growth rings the same way a tree does. So we can date mussels and relate that back to climatic conditions, which is pretty cool. But so back to the life cycle. So one of the unique features about freshwater mussels relative to those other mollusks that you saw is that they have this challenge that they live in often a unidirectional flowing environment. So they live in a stream and the water goes one way. So how do you ever move upstream? It's really hard if this is your only way of moving, which is a little foot, pseudo foot that crawls its way through. Mussels are very bad at crawling. They can move some, but very poor. And so they've developed these adaptations to help them get upstream or recolonize habitat. So the way that it works is that a male mussel will release his gametes into the water, and the female is actually going to filter them out. She has these big gills, 4 gill flaps inside of her. She's going to filter that out, and she's moved her eggs from her reproductive organs to her gills, which is really a weird adaptation. If you can imagine that in humans, that would be very strange. But so she'll brood her fertilized eggs inside her marsupial gills, is what we call them. And that's where they develop into the larval stage. So again, as a mollusk, it has a larval stage. So that larval stage is what looks like this little clam here. It's called a glochidium. And a glochidium, actually, when it was first identified, way back in the day, people thought it was like some other weird creature. It took a long time for them to connect that. A glochidium is the larval stage of an adult mussel because they look somewhat similar, but still quite different. The larval stage lacks the foot, it lacks the gills, it lacks all those adult parts that help them live in the bottom of the stream. What they are good for is latching on the fish. And so this is that really unique adaptation. So it's thought that this ability to attach to a fish originally emerged as a larval stage that had a little dangling thread that would get caught up in a fish or something like that. And over millions of years, so this is like a 200 million year relationship between mussels and fish. Over those millions of years, it evolved from a simple entanglement to actually the mussel becoming a parasite of the fish. So now this glochidium will actually attach to the gills of the fish, and if they take a bite, they get blood from the fish. But then they're quickly encysted by the fish's immune system. Basically, those epithelial cells cover it up. And then they hang out on the fish basically for a few weeks as they start to metamorphose. And once they've developed more adult features, they'll drop off. So they'll have a rudimentary foot, the beginnings of the gills, and they'll start to grow an adult shell. And then they drop into the bottom of the stream and they start to grow, and they continue to grow, sometimes very slowly, for 100 years.
So that's how they get upstream.
That's how they get upstream. So yes, if you're a mussel and you want to move upstream, you catch a ride on a fish. So it's really incredible, and there are specific adaptations. So fish are able to fight this infection. You know, fish can just slough off mussels, but in some cases, it's like this arms race between the mussel and the fish, where the mussel is escaping the immune system of the fish and is able to hang on long enough to reproduce. So some species of mussels rely on specific fish. If they land on the wrong fish, they're not going to turn into an adult. Yeah.
I think you just answered my question, but does the fish benefit at all from the parasitic relationships?
So this is a really interesting part of the discussion, because at first glance, you would think, you know, the fish really, it has just a cost from being this host for the mussel. What really, you have to think big picture, it is a parasitic relationship. So the mussel is getting something from the fish. This is really like any other irritant that the fish would encounter. So typically, unless you had some crazy infestation level in like a hatchery situation, fish don't really have a bad reaction to this. They have evolved to handle it. Where the fish benefit is later in life and how the mussel is improving the stream. So I'll talk about that. So you have to think big, but yes, they're very good for fish.
So this can be a problem in hatcheries then?
It's not really, you know, there's been maybe one example where someone tested to see how many glochidia you would need to kill a hatchery fish. You know, people love to figure out what it takes to kill something else, but we never hear of that.
And then they are host specific. They like some
Yep.
species of fish other than.
Yep, so this family, so there's two families of mussels found in North America. This family, they're the pearl mussels is what they're called. They can produce pearls, but not harvestable. And they are uniquely adapted to salmonids, which is why some of those older species are found in those cold rivers with salmon runs, for example. Other species that are in the region use other host fish. So Western ridge mussel that I'll show a picture of uses open as their host fish, which was, I believe, the fish in that first image. So yeah, so it is a unique adaptation to these specific fish. And I'm happy to talk more about that if that's of interest. But what's really fun about mussels is that there's this challenge of how do you get the fish to be there at the right time? Because as soon as you release your babies, and to be clear, mussels do not have eyes. They cannot see things. So they have to sense that it is the right time to release their offspring. They have these really cool adaptations where they actually go fishing. They attract fish to them. So I'll talk a little bit about how it's different for our species here in the Western US, but here's a few examples of mussels fishing for fish. And these are species native to the Eastern US. So this is an example of a lampsalis attracting a bass. And I might pause it to point out, but what you can see is it actually has a lure made of mantle tissue that it's waving. It looks like the type of fish that this bass would feed on. And when the fish attacks it, the mussel releases the glochidia from the gills and they attach to the gill. They're encysted there, so white. So it's not continuing to feed on the fish. It's just now kind of a passive, friend for the ride. And then they'll insist off of it once they've developed their adult characteristics. So I'll show that again. And I just want to credit Chris Barnhart from Missouri who's done this. But so you can see very quickly there, that lure that it has what looks like an eye, and it's being waved. The mussel is attracting fish. Yeah. And this has evolved, again, over millions of years for this fish to be attracted to the mussel so that there's a higher chance, because as soon as those babies are released into the water, it starts washing downstream. It's really important. Okay, here's another fun one. This is where the mussel actually grabs the fish. So it has a little lure that you can't see on the mantle pad. So the fish came up to check it out. And the mussel actually grabs the fish.
But just to release some eggs.
Yep. So the larvae are being inoculated onto the fish right now. And you'll start to see something puffing out. Those are the lochidia being released. The fish are checking it out. And then the mussel, I think it's cute. It even uses this mantle tissue to like soften. You can see how it got thicker. It's gonna soften. It's not clamping so hard that it's gonna hurt the fish. And then it just lets it go. I'm freaked out. The fish is actually gonna swim right back.
Do it again.
Emily, were these videos taken in a lab, I suspect?
Yep.
Yeah.
So Chris Barnhart from Missouri State, he has done a lot of filming and photography, and he has what's called the Unio Gallery that you can check out more.
Okay, thanks.
Okay, here's one more. This is more similar to probably what happens with our native species. So this mussel is actually going to be releasing balloons from that X current opening there. So we'll see it in just a minute. So there goes, it releases. And so we see this with Western pearl shell. They release sort of a fluff of pre-glutinate, and the fish will bite it thinking that it's food, and then it attaches to the heel. So mussels are doing lots of interesting things to get fish interested. Okay, so yeah.
Do some of those little larvae actually go into the fish's digestive system, and can the fish benefit from that nutrition?
Oh, that's a great question. No one's ever asked that. I would imagine that it's probably limited in nutrition. But yeah, it's a good question. Yeah. So this is the part about importance of freshwater mussels. So there's been a lot of research to understand, why freshwater mussels are important indicators of healthy streams. And so one of the main things is that they filter pollutants from the water. So if you have freshwater mussels filtering your drinking water, consider yourself lucky, because they remove a lot of things that treatment plants do not pull out of the water. So mussels are able to remove things like E. coli from rivers. They can actually feed on it. They remove pollutants like pharmaceuticals or personal care products, flame retardants. There's a lot of research that's shown all of the things that mussels will remove. They can actually pull DDT out of the water and store it in their shells. So they have a lot of benefits for water quality. They're also doing other neat things that they filter, which I'll talk about in the fourth panel. But they also create habitat by increasing benthic complexity. So if you think about a river with just a sandy bottom, not much ever lives in those kind of places. The water is just going to move that sediment pretty frequently, and it's hard for things to grow. But when you have freshwater mussels, it's almost like a reef system where those shells can persist for decades. They're often very thick calcium. And so they become habitat both when they're alive and when they're dead. This example is of a crayfish inside a Western Ridge mussel shell from Smith Rock State Park. And I like to show that one because I picked up like 4 of these, and every single one had a crayfish in it, so I stopped picking them up. They're clearly homes for somebody. But also things grow on the outside, they grow on the inside, they're their own ecosystems, and they're also having actual hydraulic changes on stream bottoms by basically anchoring that sediment. They can rework it with their feet and a foot, and so that helps to oxygenate the bottom sediments and loosen them up. So lots of benefits as far as habitat goes. And then they're also food for creatures like otters or raccoons. This is a nice example of a floater mussel, and I like to show this picture because I didn't stage it. There's an otter paw right there, but I swear I found a shell right next to it. And you can see there's a tooth mark on one shell and a ripped shell on the other side. And that's a really characteristic thing to find when they've been predated. And you can find otter middens that are just piles and piles of shells. They're very reliable food source because the mussel bed is going to just be in the same place all the time. So if they're hungry, they just keep going back. And then as far as their effects on other aquatic species, they actually can create fish food essentially. So mussels have this really, neat trait that they filter things from the water, but even the things that they don't want, they'll package up in mucus and they'll spit it back out. And so then it becomes this like food package for a grazer, like a snail to come over and feed off of. And so there are studies that have shown an increase in the abundance of other aquatic invertebrates that are fish food when you have a mussel bed. We also know that lamprey aniseeds can grow faster when in the presence of a freshwater mussel bed. So they're beneficial to other fish. And then there's been some recent studies that have shown that, in drying pools during summer months, fish can actually have higher survival if they're found in the same pool as freshwater mussels. And in part, that's because they're helping to keep that water filtered. You can imagine if an algae bloom started to form in kind of a stagnant pool. Well, algae is food for mussels. So they're benefiting all of the species in the river system in a kind of interesting variety of ways. They're also culturally important. Mussels have been an important food source for people as well, especially for Columbia Plateau. There are numerous examples of shell middens for especially Western pearl shell. That was an important food item. And they're considered one of the first foods. And so harvest of mussels at one time is really important, not just for food, but also for things like trade. So this is a necklace from the Confederated Tribes of the Umatilla Indian Reservation, and each one of those pieces is from a Western pearl shell. So there's been a long history of human use of freshwater mussels in the region. And then there's also some really cool examples of more recent investments by people in freshwater mussel conservation. So in the northeastern US, people understand that freshwater mussels have been really important to maintain good water quality. And those mussels have been lost from streams, so they're actually rearing mussels to return back to the rivers to improve water quality. So rather than pay for another treatment plant, they're going to try to take a more holistic approach, which is really cool. And I just have an example of a biologist in the top right who put several Western pearl shell mussels in the tank on the left, and then left the river water on the right. This is from a river up in Washington. And you can see that even after just a few hours, the clarity really changes in the tank that has mussels. So in real time, they're really making a difference for water quality. And I've got another video from Chris Barnhart's lab for that. So this is where he is using a pipette to place red dye in front of the in-current opening of the mussel. So that's where those little flecks are. The excurrent opening is behind it. And what you'll see is that this mussel, even though you can't really see it without the dye, it's constantly filtering. And so you start to see that red dye shooting out of the excurrent aperture. So mussels are filtering gallons of water a day, every day of every year for hundreds of years. Especially over a mussel bed that could span thousands of mussels and could have persisted in that spot for, such a long time. So really the capacity of mussels to improve water quality is just incredible. In Western North America, we have, again, several different kinds of mussels. I keep talking about Western pearl shell, but I will introduce some of the others too. So this is a nice Western pearl shell bed here, and I'll talk about that picture in a moment. But the map at right shows the distribution of all the species of freshwater mussels across Western North America, as far back as we have been able to put a dot on the map. So we know that freshwater mussels have been found as far as northern Mexico. There are other species found further south in Mexico, but there's a bit of a gap in distribution. But our species have been found in northern Mexico all the way up into Alaska. And in fact, one of our species is found in Russia as well. So it's kind spanning continents in the northern part of the hemisphere. What's a little bit misleading about this map is that many of these dots are places where mussels no longer occur. So for example, in Arizona, you could probably erase every dot on the map there, they've disappeared. Same for Southern California, and then various places throughout. So mussels need fish, as we know, and it's important to remember that where they are, they're present year round. So when we think about how we interact with our rivers, especially things like in-water work seasons for restoration, it doesn't really matter what time of year it is, mussels are always present. So that's one thing to keep in mind whenever working on a project that might include dewatering. Mussels can be really sensitive to that. And they don't move much, like we said, they thrive in really stable microhabitats. So like in this image, you can see that there's like this big boulder next to them, and mussels are often found in those tiny areas with a little bit more stability, which helps them stay rooted in place. That stability could be like the riparian vegetation in the first image. But so stability is really important for them. Even if we don't want a whole stabilized stream, we also do want some areas where it's a little stable for them. And they're well camouflaged, which you wouldn't think so if you had perfectly crystal clear water and a great camera, like in this picture, but I'll show a picture later. So these are the species I've been talking about. Western pearl shell. I mentioned already that's long lifespan. In that map, it's the species that has the widest distribution. So it's found up in Alaska. It used to be found in Southern California. It's found all the way into Montana. It's even in Utah. So it has a really wide distribution, but some really declining populations like in Montana. And it can be really abundant. So in places like the Willamette River, I've seen mussel beds of hundreds of thousands of them in really good habitat. Floaters are a tricky group to identify. There's a couple species that look really similar. And so to really know which species you have in hand, you have to do genetic testing, but we can usually have an educated guess. So we have species like the Oregon floater, the California floater, winged floater, and Western floater, but maybe there's only two species there. And then there's also the Yukon floater, which is in a different genus, and that one's found in Alaska. They have a shorter lifespan, just 10 to 20 years, but it can take them a while to reach sexual maturity, like 7 to 10 years in some cases with these mussels. So it does take a little while for them to become really an adult. This one is what we call a host fish generalist. So it can't use every species of fish. There are still species that do not work for them, but they can use a wider range than the Western pearl shell can, or the Western ridge mussel. So we know that they can use some salmon, they can use dace and sculpin, they can use some non-native species as well. So they're a little bit more adaptable, and they tend to be found in some of the slower moving water, more ponded habitats. They can be found in lakes as well. The Western ridge mussel, so this is a really neat species that can live up to or maybe exceeding 60 years. We really only know that it frequently uses sculpin, but it could potentially use other host fish. There needs to be more research. This species has a much smaller distribution. So for floaters, I didn't mention that they can be found from California to Alaska, as well as far east as the Continental Divide. Although I should mention places like Utah and Nevada, most of those states don't have water or mussels. So huge gaps in that distribution. But this species it kind of has its core range in Oregon and Washington, but it can be found in fringe states as well, including California, Nevada, and Idaho. And so this species has suffered the greatest declines of our native mussels in the West, and so it is currently undergoing review for endangered species listing. So I just wanted to give a nice overview of what we know about freshwater mussel distribution in the area. So I expanded beyond the Hood River area. So each of these dots on the map represents a location where freshwater mussels have been reported and we're aware of them. I listed this rivers and streams and lakes at the right. There's a lot of empty space on the map, and in fact, I only just learned about this observation in 15 miles a couple of weeks ago. It was a Forest Service observation, so that was great to hear. But we're slowly filling in the gaps. There is probably a good reason to believe that mussels are not broadly distributed in the area around Mount Hood, just because it's very dynamic. It has a very recent history of huge changes. And also glacial influence can be tricky for mussels. If you think about, I've done surveys in the White River, and there's no mussels in the White River. And you could imagine, because the filtering during those periods where it turns white must be horrible for anything with gills. So there's probably good portions of the map that just don't have mussels, but it's clear that we don't know much about their distribution. And these dots represent an observation that a mussel exists there. We don't know anything about population sizes or how healthy those populations are. So there's a lot more that we want to learn. I mean, many of these are points where someone just found a shell, so might not even represent a live mussel, but we are starting to fill in the gaps. So the observations in the middle Klickitat watershed, those are pretty recent that someone has reported them. But yeah, lots of gaps out there. So if you're out in streams looking, you could add a lot more information to this map than we have now. So we talked about the benefits of mussels, and on another slide, I did talk a bit about these same concepts. But just to kind of reiterate, and also a nice chance to see the different sorts of habitats mussels live in. They can live in boulder or cobble habitat. They can live in really like rooted networks of aquatic vegetation. They're great in sand and gravel. So there's not really like a substrate that mussels like. When people ask, well, what's mussel habitat? It's like, well, it has water, and if fish was there, and I don't know, let's go look, because it's really varied. But you know, we do know that they can't live in bedrock because they can't stick their foot down into bedrock. But if there's burrowing substrate, like a hole in the bedrock that sand has collected in, I have found mussels in that. So it's just really challenging to predict where mussels will occur. But things that we know they need to help us clue in on where to look are things like water that's present year round, some kind of stabilizing habitat features, at least a little bit of burrowing material, and fish have to be present. So freshwater mussels are really an interesting species group in North America because it's a global hotspot for these species. So more species are found in North America than anywhere else in the world. So if there's 900 plus species of freshwater mussels, we used to have 300 of them, so almost a third. Most of that diversity is not found, you know, in our region, but is really concentrated in especially the southeastern United States, where you could have 50 species in a single drainage system like the Tennessee River. So lots of diversity back east. But freshwater mussels and combined with freshwater snails are the most imperiled wildlife in North America and globally. You know, face some of the biggest decline with 30 plus species extinct in North America alone. More than 90 of them are federally listed already, and there's lots of programs to propagate them and try to restore those populations. More than 60 freshwater snail species have gone extinct. So really imperiled group. They've been heavily impacted by what we've done to our rivers and streams. And so some examples of that, some mussels used to be harvested. Let's see, is anyone wearing, you're wearing a button-up shirt, you're right. So do your buttons, why do they look the way they do? Why do they have that kind of coloration?
They call them pearl buttons. Yeah.
They're pearl buttons, right? So somebody invented a punch system to punch those pearl buttons out of a mussel shell, and they harvested mussels by the billions on barges for the purposes of looking for pearls, punching out buttons, and potentially food as well. So mussels have been harvested quite heavily, not managed in good resource, as a good resource. Habitat destruction has also, of course, played a huge role back east. There were multiple species of mussels that went extinct as a result of dams being built in their habitat, and that both affected the mussels and their host fish. So a lot of them are, reliant on a single species that might need a certain habitat too. And so declines of fish have also been mirrored in mussels as well. We have these chronic threats that we are always dealing with, habitat alteration, how we manage water, and the introduction of invasive species like the zebra mussel into the Great Lakes had devastating impacts on native mollusks. And then with recent issues, we are seeing what we call enigmatic declines, where mussels are disappearing from the stream, even though the fish are still doing okay and insects are doing okay, but for whatever reason, mussels are disappearing. And this has been really well documented in the eastern US, in Kentucky in particular. We also have a couple of really concerning issues with mass die-offs. And one example of this is at Smith Rock State Park in the Crooked River there. If you walk over the footbridge and look down, you'll see thousands of mussel shells on the bottom. And that's from an event that happened probably around 2012 or so. Totally unexplained. We don't know what killed them, but thousands of them died very quickly. And we've seen that in other places as well. I've been working with researchers to collect mussels and try to sample them for diseases to see if they might have gotten sick. But so far we don't really have any leads on what's happened. So that's a real concern because what looks like a healthy population one day could die by the thousands the next. So really critical that we understand what's going on there. There's also a few examples where mussel beds are not reproducing. So I think of it like an aspen forest. You can see what looks like a great aspen grove, tall, beautiful trees. But if you don't see the little suckers, the different age classes, that aspen grove is sort of on its way out. And we see that with freshwater mussels too, where you might have mussels all of one size class, but no babies. And so there's a great paper from California where a researcher compared two rivers, and in one river, normal, looked like reproduction maybe every year or every other year because you get mussels of the right sizes. And in an adjacent river, no reproduction in the last 30 years. So confusing, to know what's going on there. So some really important research questions that we need to answer to help conserve them. And then climate change can have real impacts on mussels, in particular with warming streams making, lower dissolved oxygen levels, the chance that your host fish maybe isn't present at the time that you're releasing your conglutinates. Lots of ways that things can get off kilter as temperatures increase. Oh, and I don't know why one of these images isn't showing. It did on my screen, but anyway, in the image on the left, I have an example of it's actually the South Fork John Day River, which was a study site of mine the last few years. There were freshwater mussel beds kind of scattered throughout the river, and the last 2 years, the river's dried up for miles in the late season. And so in 2021, I documented some mussel beds, and they were killed by 2022 because they were totally exposed. So that was a picture of that dry stream bed. So drying streams are a real concern, especially in portions of the state that are still in pretty high drought risk. There's also concerns about these wildfires. You can see that this picture is from the fire a couple of years ago in the Tahoe area that torched the forest. And this is adjacent to a site that mussels had been planted as part of a mitigation project. So can have real impacts on the stream chemistry that could affect mussels as well. And then the image on the right is from the recent debris flow in the Klamath River. This is from last fall. The area that you can see the fish kill here, that's actually also an area where we had documented freshwater mussels. And so I have not been back since the debris flow, so I don't know how the mussels fared, but you could imagine under conditions like that, mussels are not going to do very well. So they're facing a lot of challenges, and they're already in decline. And so it's really important that we recognize that they're present and that they are providing a benefit to our ecosystem. So this is the example of, I showed you the nice big mussel bed where it was easy to see them, but there is a mussel in here. And if you're a stream surveyor, you have to be really trained to pick these out as you're covering a lot of ground. So everybody sees the crayfish, right? Yeah. And so the freshwater mussel, there's western pearl shell tucked right next to it in between the rocks, but it's the same color as the rocks. It's been crawled all over by snails. You can see the little snail trails. Often there's sediment. So this is a risk for them that they are easily overlooked. So part of what we need to do is really make a concerted effort to recognize they could be present, look for them, and consider them when we're doing projects or making plans for restoration, because they're so important, but man, it sure is easy to miss them. So at Xerces, we
Emily,
yes.
somebody might have a question. Could you go ahead and ask to them if they want to unmute?
Oh, yeah, sure. Yeah, if anyone wants to ask their question online.
We'll catch them at the end.
So at Xerces, we have developed this conservation program focused on educating people about mussels. So thanks for inviting me. But also trying to put out resources and actually assist on the ground. So we do things like go out and do snorkel surveys to help identify if mussels are present. We can help move them out of areas where restoration is happening. We host trainings on how to identify them and understand more about them and their habitat. And we put out resources. I did bring some examples that these are all available on our website, but yeah, we can pass these around. We have a brochure, we have a mussel ID guide, and then BMPs. But those are all available online, and they're intended to help people understand how to search for mussels. We can also provide that distribution data if you need it for your projects. And we'd love it if you share distribution data, because then we can share it with agencies and other organizations as well. But our goal really is to just make sure more people are aware of mussels and try to help understand where they are so that we can find solutions to the declines. And just one really fun example is this monitoring project that I've been involved in on Murderers Creek, which is a tributary to the South Fork Down Bay. This is an example where we're trying to understand how mussels respond to fish habitat restoration projects. So we have pit tagged 6,000 mussels by painting glue, sticking a pit tag on, covering it with epoxy, and then putting the mussels back in the water where they burrow in quite happily, and then going out and using a pit tag reader to try to find them later to see if they got washed out or find shells that have been predated. For example, we do find a lot of predation there too. But so this is just an example of some of the new things that we can do to kind of better understand how we're impacting mussel populations, both for good and bad.
When will the results of that study be done?
2026.
Okay.
Yeah, we're trying to do it multiple years before the restoration, which is happening this summer, and then multiple after. So, yeah, but we'll probably put out some interim results too.
And are you salvaging mussels for the restoration project as well?
We are.
Making sure that they're not,
Yeah.
relocating. Yeah.
Yeah, because part of the project includes like adding gravels back to the stream, and so if you put gravel on top of the mussel bed, they have a hard time unburrowing from that. They're just not that strong. So, so yeah, we're going to definitely move those guys. So just the question or people get really excited after we talk about mussels, which is cool. So it's just important to note that mussels are protected in a couple ways in Oregon and in Washington. So you do need a permit to handle them. You can add them really easily onto a scientific collection permit or a permit that you might need for fish salvage, for example. But we don't really encourage picking up live mussels. But you can pick up the shells, you can collect them and carry around a bucket like I do. And it's also important to note that harvest of them is restricted in the fishing guidelines. Unfortunately, there are examples where people advertise cooking Western pearl shell mussel, but the punchline is that they really don't taste great. They are an important food source. If you need food during lean times, you will live off of the mussel, but you will not like it. So, yeah.
You mentioned they're good at filtering pollutants, including man-made pollutants. If they're filtering man-made pollutants, does that make them toxic for human consumption?
So they're not toxic in the way that if you, for example, eat a razor clam that has been exposed to some of those algae that create biotoxins. We don't really see that in freshwater systems, but certainly because they are collecting a lot of nasty stuff in their tissues, you wouldn't want to eat them. And there have been studies that have looked at, there was one on the Klamath River that the Creek tribe was involved in, looking at maybe setting a standard for how many mussel fillets or whatever you would call it, you could eat over a certain amount of time. And I think they really came up with, they need more information to set that, but yeah, I wouldn't recommend it. And honestly, you know, like this was a example of illegal harvest from the Willamette River. And the guy posted online at that website. And in his review, he said, no matter how much butter and salt I added to it, it still tasted terrible. So he was saying, okay, well, you just killed, you know, how many mussels in there for a meal you probably threw away. So anyway, it's not legal unless you have a reserve treaty right. So like the Creek tribe and C2IR both have used mussels, but the tribes also recognize that the mussel populations are declining, and so really don't have active harvest of any kind. Or any significant amount. So, but if you do want to, you know, share observations of mussels, like I said, that's really valuable. There is a that link, the ArcGIS link there is to a survey 123. So if you ever use that, you can quickly document observations of mussels. You can also use iNaturalist. There is a project on there called Freshwater Mussels of the Western US. And so you can share those observations or get in touch with us at Xerces. But that kind of information is really valuable. And in fact, you know, I recently heard about an observation of floater mussels in the island. I forget the name of it, but the island that's just off the hook. Yeah, I think someone was mentioning they saw live floater mussels around there. So yeah, so it's great for people to share that. So anyway, I just want to take a moment to thank many of the organizations that have supported our work. We're grant funded, like you all. So just important to acknowledge how much they've contributed to this. And then I'll finish with my current favorite mussel bed that I visit every year and go look at. But a really gorgeous photo from Roger Tabor Fish and Wildlife Service in Washington, but a really abundant Western pearl shell bed, probably 20,000 mussels in an area a little smaller than this room in the river. So really, really great to see some of these riding beds out there. So
on their distribution, what about depth? How deep?
Great question. Yeah, they can be really deep. So so mussels don't need sunlight, right? So I find them under boulders. They can be really deep in the water, like 30, 40 feet down. I have seen it's really cool in the Smith River in Northern California. That's a really clear river that you can see way down, and there's pockets where I can see there are mussels like 30 feet down, but couldn't get to them. So yeah, they seem to do well with depth. Okay, so so the question is about determining which species from the glochidial stage, and yeah, the size and the shape of that larval stage when it's attached to a fish, you can identify which species it is. So the floater mussel is more triangular. The Western pearl shell is going to be a lot smaller, and then process of elimination, the other one is Western ridge. So that's a great question. Another good question about the freezing during very low flow events and cold temperatures. Could that explain some of the die-offs? Yes. So I have seen examples where after the winter, there will be lots of shells. You know, something killed them over winter. And one of the theories is that anchor ice forms on the bottom of the stream and then scours them out and can freeze them as well. I don't think anybody's ever actually documented it, but that's definitely come up as a theory a couple times.
What about upper lethal temperatures in this water?
Yeah, we don't know what they are for our Western species yet, but there's a graduate student studying that for the Western ridge mussel currently. And people have studied that in Texas recently as well, and I think their conclusion was that it's water temperatures there are already reaching thermal limits. So yeah, so they seem to be able to tolerate warmer water than salmon would, but beyond that, no. Yeah.
Okay, so, how long, the local mussels, how long can it stand dewatering?
Yeah. Oh, yeah, that's a tricky question. It really depends a lot on the species and the habitat. So my experience has been, I have seen floater mussels that have died within hours in a place like Oaks Bottom, where there was a really sudden drop in the water. And they were in this clay habitat, and I guess the clay just doesn't hold hyporheic flow. So they were exposed without water to the air, and it was a hot day. And also like predators were showing up. That's where that predator picture came from. There are other instances, other species of mussels like back east, people have found that they can survive in the hyporheic zone, where as long as it's still wet somewhere, they can go for a couple months where the stream looks dry, but they're under it. And I didn't talk about their burrowing, but mussels do burrow up and down in the stream, often seasonally, if they're in habitat that allows for it. So like cobble habitat, that's really hard to do. So that's another example, is like in cobble habitat, they probably die pretty quickly. Yeah, but as for like, you know, it's so temperature and condition dependent, I would say, but pretty quick.
I hear about ocean acidification hurting the growth of ocean shellfish. Do we observe acidification of freshwater habitats and the effects on freshwater shellfish?
So in the eastern US, I know there have been a couple of projects to add lime back into streams to help buffer the streams. And so I know that's a practice that has been done. In the western US, we do see examples. So mussel shells are composed of 3 layers. The outside layer is protein, and I'll point you that in just a minute. But the outside layer is protein, and then there's a calcium carbonate, like really thick layer, and then the mother of pearl, which is also calcium on the inside. And the protein layer is really easily lost or dissolved. And so that can often be the first thing to disappear. So we do have examples where people find shells and it's like the protein layer has just been almost completely dissolved. And then, you know, then the calcium layer becomes more exposed. And so mussels can rebuild their shells to some degree. They can hatch things. That's how we get pearls, for example, but their ability to kind of withstand more acidic streams is probably not great, but it might affect internally more than externally first. I'm not sure. Yeah, so specific pollutants that are known. So ammonia is like the real killer of mussels that we've identified. So they do really poorly with ammonia, which is why the EPA developed a water quality standard specific to freshwater snails and mussels, because it was shown that they really are like the least tolerant thing in the stream. So anywhere where there's a wastewater treatment plant with a pipe, they have to account for whether mussels are present or not as to how much treatment they have to do now. That's the main one. They seem to be less impacted by pesticides that have been tested so far, but heavy metals can also be damaging to them, so like copper in the issue. Thank you for being right. Yeah. Just call it out. pH. Is there a specific pH? I don't know the answer to that. I'm not sure how low of a pH they can tolerate.
I'm going to stop.
Yeah.
Regarding harvest regulations, are there exceptions for non-native species, and are any non-native species at all?
The corbicula, the clam that I showed at the beginning, there's some thought that maybe it was introduced on purpose as a food source. So I do know that like the city of Portland released a warning for within the downtown area of not eating them because they also can have high level of contamination, but I do know that they are used for food in some instances. And they're also food for species like sturgeon, actually, in the river, feed on them. And they're really the only non-native bivalve that we have currently. We do have other non-native snails.
Do the regulations allow harvest of the non-natives, setting aside the?
Yeah.
They do.
Yeah, they do. Yeah, it really just restricts the freshwater mussels.
Yes, so I'm now kind of curious. So you know how we do, all kinds of stuff in the stream, whether it's flying surveys or, fishing or just. How do they withstand like just being kind of trod on
Stepped
really
on.
quickly?
Yeah,
Like without even knowing that they were there.
yeah. No, it's a great question that I often think about and constantly imagine, how many mussels are just swimming in, no. And again, it's kind of habitat dependent, so like kind of the same as well, if you think about it, like walking in a stream that has a lot of cobble, probably most of your weight is resting on the rocks themselves and the mussels are between them. We do have instances in places where we have, for example, gone in to collect mussels and move them out of a restoration site, and from walking back and forth, we find mussels that have been stepped on and their shells have cracked. But I would say that my experience in that big pit tagging project, we encountered so many mussels, tens of thousands in that project. And I've really only seen evidence myself that maybe 4 or 5 of them had been cracked. And I've revisited those locations a lot. So I think really the stream environment is pretty forgiving as far as walking. And we find them in streams where spawning surveys happen every year. So yeah. So don't worry too much. Don't let it keep going. So if there aren't any other questions, I can pass around a couple of shells, just so you can see.
Yeah, I think you might be able to show some. This is the can here, I believe. Yeah, that lit up
Yeah.
spot there.
Yeah, so
Already memorized it.
so I always like to bring a couple of examples of things we find in streams that are not freshwater mussels. So razor clams are really commonly found, especially around boat docks. We also find oysters. This was from like a coastal stream. So if you find a shell that looks really weird, it might not be from the place you found it. That's a good thing to keep in mind. But I'll pass around first. This is the corbicula. So this is, they can get bigger than this, but this is a pretty standard size. One of the things to notice as you handle the shell is that if you look at it from the side, it almost looks like a heart shape. So the umbo is the oldest part of the shell, and it actually sticks up above the hinge that connects the 2 valves. And so when you see a heart shape like that, it's not a native freshwater mussel.
It's not.
I'm just
It's
gonna
not native.
hold that up closer there.
Yeah.
Can you guys see that better?
Probably is full of those
Yeah.
sturgeon. Eat them all in the spring,
Yeah, and important.
Sturgeon love those.
All right, let's try to. Okay, we got it.
Yeah, so you can see that heart shape.
Hey,
Everybody's still.
there we go. Yep.
but-
Don't talk. It's
Don't fucking stay still.
it is the.
So another example is the Western Ridge mussel, often has an actual ridge that you can feel running from that hinge line all the way down to the point. So that's an easier one to identify. Let's see, I can get it.
Where in the West do you find the most diversity, like a stream system or whatever that has the most diversity of mussels?
Hey. Thanks, Megan. Oh, there we go.
Here's the state and the Western Ridge. The greatest diversity seems to be in places like the Columbia River, Maine that has seems to have all the species. Klamath River also has species. And there's, it does, yeah, and then there's a few others as well. Yeah. Okay, so this is a super monster beefy Western pearl shell. You can really feel the heft on that. They often lay down a lot more calcium in their shell than other species, especially compared to the floater, which obviously different sizes, but this is a floater mussel, which is going to be a lot more lighter weight. So,
All right, Western pearl shell people.
give you a coverage. This is a really
Western, Western, this one's a Western pearl shell that I'm holding up again.
Yeah, that's like running away.
And this is the
Name.
floater, of half of the floater.
Yep.
That's it.
Yeah, so the floater mussel,
Yeah,
the thought is that the shell will float after it dies, whereas the other shells. You're holding Western mussel shell. Both. Yeah, so yeah, they have different shapes depending on the habitat.
Most common or is the lead?
Um,
Like on the.
yeah, it's so hard to know because, I haven't been able to do surveys really of why, yeah, because we need people with that. Yeah. Yeah. So they don't even fit. They're shorts. The particulate, yeah, but. This is a floater. This is what we.
Oh, this
They do, yeah.
These
So
don't-
here's the bigger one.
They don't seem thick enough to make buttons out of. That must have been-
Yeah. And they found that after the process of
These are both Western pearls?
Western Pearl shell. So they just can have really different shapes depending on the habitat. Those are around here in Western River. Yeah.
This one may have been fire.
Okay, I'm gonna butt in here first. I think for the sake of folks online and whatever we should, first of all, we really thank you, Emily, for that wonderful presentation, and here's a round of applause. And so, there may be more activity around here in the room with looking gels. It looks like we're slowed down on that. So if anyone doesn't have any other further questions, I will say that our presentation and watershed group meeting are over. So if you have something, shout it out before you.
I think that's it.
Okay.
Thanks all you online participants.
Yes, thank you.
Yeah.